Chromatography is a laboratory technique that used for the separation of the mixture into its individual components. The basic principle involves passing a mixture dissolved in a mobile phase through the stationary phase. There are types of chromatography, which is paper chromatography, thin layer chromatography, gas chromatography, liquid chromatography, and column chromatography.
Here are some examples of food dyes. Red 40 known as Allura Red AC and Yellow 5 known as Tartazine. Both of these are synthetic food dyes. Moving on to the natural food dyes, which is annatto from achiote seed and chlorophyll from green plants. Now are the example of candies and snacks that have food dyes. For candies, we have Skittles, which contain red 40, yellow 5, and blue 1. Next is Starburst, which have red 40, yellow 5, and yellow 6. Moving on to the snacks, we have Cheetos, which have yellow 6, and Cheez It, which have annatto.
So next we are moving to the how does chromatography detecting food dyes in snacks and candies. First, we have sample preparation, where we have extraction, where this process is the dye is extracted from from the food sample, and this is usually involves dissolving a portion to the snack or candy in a suitable solvent. Second, we have filtration, where the solution is often filtered to remove any solid particles. Therefore, we have the food sample that we need to be before we doing the chromatography. Next, the application to the chromatography system, where this step is where we are going to separate the dyes of the food sample to determine what is contained in the food dyes. So for the thin layer chromatography, the process is a small drop of the extract is placed on a TLC plate. Meanwhile, for the high performance liquid chromatography, the extract is injected into the HPLC system. Third, the gas chromatography, where the extract is vaporized and injected into the GC system. Therefore, each chromatography system have different of inserting uh, the 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 food dyes sample. Number 3, we have the separation process, where in thin layer chromatography, the TLC plate is placed in a solvent chamber. As the solvent rises up the plate by capillary action, it carries the dyes with it, separating them based on their affinity to the stationary phase and the solvent. For the HPLC, the sample passes through a column filled with a stationary phase. Under high pressure, different dyes interact differently with the stationary phase and elute at different times. For gas chromatography, the sample is carried by an inert gas through a column coated with a stationary phase. Dyes separate based on their volatility and interaction with the stationary phase. Therefore, for the separation process, we can see that the food dyes is developing and also separated truly accordingly to the chromatography that we use. Lastly, it is the detection and also the analysis, where for the thin layer chromatography, the separated dyes are visualized under UV light or by applying a suitable detecting reagent. The RF value, which is ratio of the distance traveled by the dye to the distance traveled by the solvent, helps in identifying the dye. For the HPLC, a detector, which for example UV visible detector, identifies and quantifies the dyes as they elute from the color. For the gas chromatography, a detector such as mass spectrometer analyzes the separated dyes based on their mass to charge ratio. Therefore, we can see that in detection and also analysis, This is where the process where we can see what does compound or component in the food sample that we use. For the application in food analysis where different food dyes have unique chromatographic profiles. For example, retention times in HPLC, RF values in TLC, and so on. By comparing these profiles to those of known standards, the specific dyes present in the sample can be identified. Chromatography can also determine the concentration of each dye present in the sample. Therefore, the chromatography in detecting of food dyes in snacks and candies usually go through many processes where it can be so accurate, and it can be determined with three chromatography, which is thin layer chromatography, high performance liquid chromatography, and also gas chromatography.
I will be talking about advantages using chromatography in detecting food dyes. Number one, high sensitivity and precision. That means that food dye can be accurately detected and quantified even in minutes amounts using chromatography. Number two, separation efficiency. That means that it can efficiently dissect interrelated combination into the constituent parts, simplifying the process of identifying certain dyes. And number three, versatility. That means that various types of chromatography, example, liquid chromatography, gas chromatography, and thin layer chromatography can be used depending on the nature of the food dyes and the matrix. Number four, qualitative and quantitative analysis. It's offer quantitative data figuring out how much of each dye is present, in addition to qualitative data identifying which dyes are present. And lastly, number five, non-destructive testing. That means that the material is largely intact, enabling additional testing if necessary.